My name is Constance Jeffery, and I'm from the University of Illinois at Chicago. My poster is about enzymes, moonlighting enzymes, and pseudoenzymes, proteins that are similar in sequence but different in function. This idea of having different protein functions within a protein superfamily is illustrated in Figure 1. These all represent homologous proteins, and in this protein superfamily we have an enzyme with what we call the canonical function of the protein superfamily. We might also have some additional enzymes that vary a little bit in their substrate or their catalytic mechanism, resulting in different products of the reaction. So those would be non-canonical enzymes. And we also have proteins like protein 3 here, a pseudoenzyme. Now, a pseudoenzyme resembles an enzyme, but it's missing something. It has some difference in or around the active site so that it does not have the catalytic activity. But they often retain some aspect of the catalytic activity, like they might still bind to substrate, or they might interact with other proteins, other catalytically active homologs, and regulate the function of those homologs. A protein superfamily can also include moonlighting enzymes, and these are enzymes that have an additional function, often a non-catalytic function, in addition to their catalytic function. And finally, we can have proteins that evolved from moonlighting proteins but lost either their catalytic function or the non-catalytic function. If they lost their catalytic function, they might have the properties of another pseudoenzyme, but sort of a different type of pseudoenzyme from what we usually think of as a pseudoenzyme. And these different types of pseudoenzymes can be illustrated or are illustrated in panels A, B, C, and D in figures 2 and 4. So first of all, an enzyme generally has an active site. In this cartoon, the active site is catalyzing a reaction where you're transferring a functional group from one molecule to a target molecule represented by the brown rectangle. A related pseudoenzyme might lack the catalytic activity, but it still has the binding ability to interact with that other protein. Now, a moonlighting enzyme would have a catalytic activity, and it might have a second function where it binds to a protein represented by the brown oval. But this binding function is separate from its catalytic function. A pseudoenzyme that evolved from a moonlighting enzyme would be lacking the catalytic function but would have this other protein binding function. An example of proteins like this are in the arginosuccinate lyase superfamily. Arginosuccinate lyase is a ubiquitous protein in the urea cycle. And in some species, including ducks, we find an arginosuccinate lyase active enzyme that is the same protein as a crystallin. Our crystallin is a protein that has a non-catalytic function in the lens of the eye. Also in ducks is another homolog of arginosuccinate lyase. It's another crystallin. This is the delta 1 crystallin, but this protein does not have any catalytic activity. So it's a pseudoenzyme that has 94% sequence identity to the active arginosuccinate lyase delta 2 crystallin. And in Table 1, we list several other proteins that have pseudoenzyme and moonlighting enzyme homologs. So in summary, one protein family can contain enzymes, moonlighting enzymes, and pseudoenzymes. Many pseudoenzymes have functions that share some aspect of the function of a catalytically active homolog, but other pseudoenzymes have a very different function from their catalytically active homolog. These latter proteins might have evolved from ancestral moonlighting proteins. So this suggests that comparison of proteins that we think might be pseudoenzymes, for example, proteins that resemble enzymes but lack conserved amino acids, might help us come up with a hypothesis of what these proteins do. If these pseudoenzymes resemble moonlighting proteins, we might be able to predict that the function of the pseudoenzyme is similar to that of the non-catalytic function of the moonlighting protein. Thank you for your time and interest.